Okay, I think we're all set up. Not sharing the right screen. I am sharing the right screen. Okay. So when we left off last time, we were talking about the GI system, and we got through what's going on in the stomach that regulates acid and pepsinogen secretion. And in order to make sure that we actually get through both the male and female reproductive systems, I said we're going to basically stop with the GI system and just move forward. So what I'm telling you is that bit on the pancreas and how it's regulated, we're going to skip that for now. Did say I want you to know basically 2 words that we hadn't gotten to left in the GI system. And those are segmentation and peristalsis, because we're going to see the word peristalsis again as we move into the reproductive system. So it's not helpful to us to move forward if we haven't learned this word yet. So basically what peristalsis is it's just movement in muscle, so waves of contraction that move something forward. And when we're in the GI system, we contrast this with segmentation, because when we're thinking about digestion and absorption, we also kind of like shake up the contents and kind of keep it in place without moving it forward, but mix it around so that everything comes into contact with the cells lining the gut lumen so that we have the opportunity to absorb any nutrients. Okay, so if we were to look at a picture of peristalsis, it would look something like this. So the really basic thing that's happening here, which is really what we're interested in, is that if we have like a bolus of food, but in the future we can think of this as anything we're trying to move along, we're trying to move it forward. That's peristalsis. We're moving it forward basically by contracting muscle kind of behind it, making kind of a squeeze point so the thing that we're moving doesn't move backwards. And then we kind of shorten the part in front of it by relaxing longitudinal muscles, which allows this to kind of like expand wider a little bit. And then this just kind of progressive contraction of this like pinch point moving forward from behind kind of pushes our thing forward, or in this case, our food. In the case of the male reproductive system, we'll be seeing that this happens with sperm as well. Okay. So that's peristalsis. We're just moving things forward because we're squeezing behind it. So basically, we're making this posterior region have this small diameter, which has the effect of pushing things forward. Okay. And we just want to know that peristalsis is different than segmentation, which is the other type of movement we see in the gut. So segmentation is specifically really happening in the small intestine, because that's where we're doing our absorption. And you can see from the colors bouncing back and forth, right? Like what we're actually doing is just kind of mixing our food back and forth, shaking it up a little bit so that we basically have more opportunity to come into contact with absorptive cells that would be in the walls here. And it mixes our chyme, right? We know that we have acidity coming from the stomach. We want to mix it with bicarbonate so that the contents of our intestines don't stay super acidic as they move through the small intestine into the colon. So it's important that we mix stuff up. So this happens with alternating contractions. So the difference from peristalsis is that instead of having this like wave of contraction behind that's moving our ball of food forward, because it's just moving in the same forward direction, we kind of jiggle it back and forth. So that's what's going on with these arrows, right? They're just showing you that there's contraction here, right? Moving the ball of food, and then there's contraction here, moving it backwards. So segmentation is about mixing, peristalsis is about moving things forward. It's just the difference between those two terms. So from there, we're going to move into our reproductive system. Okay. So today we'll be talking about basically how the reproductive system forms in embryology, and talking about the male reproductive system Then on Friday, today is Wednesday, on Friday we'll talk about the female reproductive system. And then this class is basically over. We'll be reviewing pretty much all of next week. We'll review on Monday for your exam on this last unit, and then we'll review for your final exam, which you should remember is worth a little bit less than a normal exam. It's 50 points, and we'll try to stay on broad topics for that, but it's been a while since we started this class, so we want to make sure we have some time to touch back on things we did much earlier in the semester. Okay, so first thing we want to get straight about our reproductive system is just a kind of basic view of what it is, what are the general things that are going to be true about both the male and female reproductive system. What are some of the factors we want to be thinking about? So we want to be thinking about terms like gametes, terms like sexual reproduction. We're not going to relearn meiosis, but we want to kind of jog our memories and remember that meiosis is a type of cell division that exists. And we're going to see how all these things kind of come into play in the male and the female reproductive system. We're also going to talk a little bit about fertilization and lifespan patterns in reproductive activity, because the reproductive system doesn't stay the same in, a little kid versus in someone who's 80, right? We know that the reproductive physiology changes over your lifetime. Okay. So we're going to start out by thinking about sex chromosomes and some sex hormones and how they lead to the development of different sexual characteristics, by which we really mean how they lead to the development of either a male or female reproductive system. Okay. So when we use this term reproductive system, we're referring to basically all of it, right? We're referring to all of the organs that are going to be involved in mating, or copulation, intercourse. We're referring to any organs that are going to be involved in gametogenesis, which is the production of sperm or ova our egg cells, or any other functions involved in producing offspring. So all those pelvic organs, pretty much, except for the bladder that you've been looking at in lab, that's our reproductive system, right? We've seen the pieces of it already. One of the things we're going to focus on is the gonads, because these are really the specialized primary sexual organs that we're going to be interested in terms of producing gametes, producing sperm and egg cells. So in males, the gonads are referred to as the testes, which you've seen in lab. In females, the gonads are referred to as ovaries. So these are kind of analogous, right? They are similar organs, but in males versus females. And their major functions are to produce gametes and to produce sex hormones. There are also accessory reproductive organs. So there are going to be other organs involved with the reproductive system that aren't directly producing sperm and egg cells. So it's not like the gonads are the only organs we're going to see, but they're going to be our focus. Okay. So one of those roles of the gonads is to produce sex hormones. So you've probably heard about some sex hormones before, right? Like testosterone, estrogen. So those are sex hormones. And actually, both males and females do have both kind of classes of sex hormones. Androgens are generally associated with males and with men, things like testosterone, right? Really mainly we mean testosterone, that's our main androgen. The reason for that is it's produced by the testes. So women don't have testes, so they do have fewer androgens, they have less testosterone, but they don't have zero because it's also produced by the adrenal cortex. So remember those adrenal glands sitting on top of the kidneys? The cortex of those glands also produce androgens, so they would be present in both males and females. Estrogens and progesterone are the typically female sex hormones, but we will see those hormones in males as well, but females have more, basically. In the accessory reproductive organs, we're going to see that their role, rather than producing sperm or ova on their own or producing these hormones, really they're going to be involved in transporting gametes or secreting different types of fluid that allow the sperm and ova to move and to do their jobs. So we'll see some of them producing enzymes, things like that. So those are the other organs and the other structures that you've seen in lab largely. We are going to spend a fair amount of time talking about the development of the gametes, so talking about the development and release of sperm cells and egg cells. But part of why we're interested in this, right, is we're interested in, how does our species reproduce, right? Like, how do babies get born? How does sexual reproduction work? So we also want to make sure that we understand copulation. So the act of actually mating in order to produce offspring. So we'll talk about how sperm get deposited. And we'll also, when we move to the female reproductive system, talk about what happens when ova become fertilized and how that would work. We'll touch briefly, again, more on Friday when we're talking about the female reproductive system, about pregnancy and gestation. So we'll see two terms when we get to that point. We see that in the first 2 months, we refer to the growing organism as an embryo. After 2 months, we refer to it as a fetus. Okay. And about 9 months in humans, right? This is a human A&P class. At about 9 months, birth happens, also more technically referred to as parturition. But that's more associated with what we're doing on Friday. Okay. So here is where I kind of want to start our conceptual view for today. So in order to understand our reproductive system, we kind of want to know what causes some people to develop a male reproductive system and what causes some people to develop a female reproductive system. Right? There's no magic wand, right? It's actually a kind of complicated process, as is the development of secondary sexual characteristics. Usually, the way it should work, or the way it works when it's working the typical way, is that chromosomes determine whether a fetus develops ovaries or testes, and then the hormones that those ovaries and testes produce leads to kind of a cascade of effects in people. So really what we focus on is the presence or absence of a Y chromosome. And the reason for that is that the Y chromosome carries a specific gene, the SRY gene, which is called this because it is the sex determining region of the Y chromosome, the SRY. And the SRY gene codes for something called testis determining factor. Well, that's that term at the bottom here, testis determining factor. And testis determining factor determines whether you will produce testes. So if you have this SRY gene, you will produce testis determining factor, which will lead to the production of testes. Okay. In the case that a Y chromosome is absent, development still happens, right? Embryology still follows a normal course. You still, develop some type of reproductive system. What you develop is a female reproductive system if this SRY gene is absent, and therefore there's no testis determining factor present. So females develop because they lack a Y chromosome, so they only have X chromosomes, right? They usually have 2 of them. So with the absence of this SRY gene, they develop ovaries. So this is all controlled by hormones. So the first kind of one that we're thinking about is that testis determining factor. But we're going to be really interested in development in the presence and absence of testosterone and another hormone that the testes create, that they secrete, called Müllerian inhibiting hormone. So these are two products of the testes themselves. So you'd have to have the SRY gene producing testis determining factor, creating these testes, and then the testes are going to secrete testosterone and Müllerian inhibiting hormone if we're going down the male route, leading to a male reproductive system. Okay. So the testosterone and the Müllerian inhibiting hormone are going to act on two structures that exist in the embryo already, that exist in every embryo, male or female. Okay. So these are called the Wolffian ducts and the Müllerian ducts, and we're going to look at them in a second. But they're basically kind of two parallel pathways that through the course of development are going to lead to either a male reproductive tract or a female reproductive tract. So what's going to happen is that if we're going down the male route, we're going to create a male reproductive system, we need to make the Müllerian ducts go away, right? We need to make them degenerate so that we're only left with Wolffian ducts to produce a male reproductive tract. Or in females, we would need the opposite thing to happen. We need to get rid of our Wolffian ducts, leaving only Müllerian ducts. And the way we kind of select these is using these hormones. So I want you to focus first on this Müllerian inhibiting hormone that's produced by the testes. So its name already tells you what it does, right? So the testes produce this hormone that inhibits, right? So that kind of helps us delete the Müllerian ducts, right? So the Müllerian inhibiting hormone inhibits the development of the Müllerian ducts, so they degenerate, they kind of collapse, kind of fade away. That leaves us with only the Wolffian duct. The Wolffian duct is stimulated, its development is stimulated and encouraged by testosterone. So that's what the testosterone is doing in the male reproductive system early on in embryology. Okay. In the case that no testes are present, because we don't have an SRY gene, because we don't have testis determining factor, what's going to happen is that the Wolffian ducts are going to regress, so they're going to degenerate naturally. This is kind of just the pre-programmed process. If nothing helps them out, the Wolffian ducts kind of die off, allowing the Müllerian ducts to develop instead. So that is what would lead to a female reproductive tract if we don't have the testosterone to stimulate further development of the Wolffian ducts and the Müllerian inhibiting hormone to kind of turn off the Müllerian duct. So you can see what that looks like in this image from your textbook. So what we're looking at here, right? So the Wolffian duct is this kind of central one. It's also sometimes referred to as the mesonephric duct. Okay, so this is the Wolffian duct. And so then out kind of lateral, but moving towards the center. This one is our Müllerian duct. Okay. So we see the difference in male and female development as we move down. So we can see that in male development, the Müllerian duct is degenerating because of that Müllerian inhibiting hormone, right? So this kind of dotted line is indicating that the Müllerian duct is going away. And we can see that the Wolffian duct is growing larger, it's changing shape, and it's enveloping early kind of glandular tissue. So kind of glands that we'll have in the center of our gonads are getting enveloped by the Wolffian duct in this case. Versus in the female development, we have the opposite thing happening. The Wolffian duct is going away. So instead, the Müllerian duct is staying put, growing a little. We can see that it's forming a uterus at its base where two Müllerian ducts come together. Our gland is staying and just forming an ovary in the female development. So over the course of an individual's lifespan, we don't just develop these reproductive tracts and then, you know, we're born and that's it. That's all that happens. We also have progressive development throughout life. So one of the main things that happens is puberty. So beginning at about 10 to 14 years of age, sexual organs begin to mature. So they start to be able, in the case of males, to produce gametes, to produce sperm, or in the case of females, to make ova, release, and develop. We'll talk about how that system is different on Friday. And this is also when secondary sexual characteristics develop, like in females, breasts and hips, in males, maybe a beard, different musculature. So characteristics that tell us visually, you know, whether we're seeing someone who's typically male or typically female, although we know a lot of variation does exist. Then during sexual maturation, so like after we have hit puberty, after puberty has kind of finished out, the female reproductive system cycles, right? There's a menstrual cycle where eggs are released on a more or less monthly basis. The male cycle doesn't happen the same way, right? Males really have a similar progress day to day, so there's no corresponding cycle in men. And another difference in sexually mature males versus females is that women go through menopause. So females lose reproductive capacity later in life, whereas males retain the ability to produce sperm throughout their lifespan. So next, we're going to talk about spermatogenesis and how hormones regulate the development of sperm. So here we see the male reproductive tract. So this should be familiar to you from anatomy lab. We can see at the bottom here, right? We have our testes. This is where our sperm are going to be produced. They're going to begin development. They'd then be released into the epididymis, travel through the vas deferens. They're then traveling kind of behind and under the bladder, joined by fluid from the seminal vesicle. They're crossing through the prostate, which is also going to add some fluid. Here I'm tracing through the ejaculatory duct. They're then going to enter the urethra, where something called the bulbourethral gland will also add a little bit of fluid. I don't think that one was on your lab list, which is why I'm pointing it out here. Then we continue through the urethra, through the erectile tissues, which is the corpus cavernosa and the corpus spongiosum, and eventually out through the exit where sperm can be deposited. So out through the external urethral orifice. So for our lecture purposes, we're going to look in kind of more microscopic detail at some of these structures, and specifically at the testes. So the testes are the male gonads, so they're producing sperm and they're producing androgens, they're producing sex hormones, in addition to producing testosterone and Müllerian inhibiting hormone during development. They do kind of their own thing during the actual lifespan as well. So where we're going to be kind of looking for our purposes is inside the testes at these structures called the seminiferous tubules. So in the testes in lab, where you can see them bisected, it just kind of looks spongy inside. But what we're really looking at is a series of coiled tubules in compartments, like almost 300 compartments. And if we look even closer, these tubules are made up of smooth muscle surrounding the tubule, and then the tubule wall is made up of these cells called Sertoli cells. And then the inside of the tubule we refer to as the lumen. So that is the structure of the seminiferous tubules. We do have one other cell type we want to be thinking about as we think about the testes and sperm development. We have the Leydig cells. So these are here in kind of purple blue. So the Leydig cells would be lying in between the seminiferous tubules, kind of in the blank space. And they're the ones that are actually secreting testosterone, which is why we want to know about them. Okay. So kind of between the seminiferous tubules, we're producing testosterone, this hormone. In the tubules is where we're going to be developing sperm. So the Sertoli cells, these cells that line our tubules, are going to be important for sperm development. So we're going to look at them more carefully as we move through the rest of today. Okay. Before we look at the specific steps of sperm development, we do kind of want to just remind ourselves of the pathway that the sperm are going to follow. So the testes, once we produce sperm, we're going to have sperm in the lumen, right, of the seminiferous tubules. So I have sperm in here. They're going to need to move forward through the entire reproductive tract, right? They're going to have to go back up into the body. They're going to have to pass through the prostate, through the urethra, and then get out the penis. So that's going to take some work. So this is where peristalsis comes back into play. This is why I wanted to make sure that we covered that term when we did GI. So just like we saw in the GI tract, there are going to be layers of smooth muscle in the testes that progressively kind of squeeze the sperm forward. So they do peristalsis. So the sperm move by peristalsis through the male reproductive tract. Another thing we want to be aware of as we're looking at the seminiferous tubules is we want to know that there's this thing called the blood-testis barrier. We can kind of think about this like we thought about the blood-brain barrier. So there's a really tight kind of separation because of tight junctions between the sort of external part of this tube, the basal compartment, which would be the part that would be nearest blood flow. Right, so we can think of this part above our tight junction as our basal compartment, versus what we're going to see in the luminal compartment and kind of nearer down here. So we have a blood-testis barrier, which means we're going to be able to very kind of carefully control the hormones and the other nutrients that are present in this area where the sperm are developing. Okay. So that brings us back to the Sertoli cells. Okay. So the Sertoli cells are the ones that are lining the tubules, right? So they're going to be in direct contact with the lumen of the seminiferous tubules. And we can see them here as well. So based on their position, shouldn't be a huge surprise that they secrete something called luminal fluid. So they're going to be secreting fluid, goes into the lumen and nourishes the sperm. Another thing that they are going to be secreting is something called androgen binding protein. So the testosterone is coming from the Leydig cells. The sperm do need testosterone to develop appropriately, but we want that level to stay steady. So androgen binding protein secreted by the Sertoli cells it's kind of acting like our bicarbonate did for acidity. It's kind of buffering the levels of androgen and making sure they stay at a steady, constant rate in the lumen that's actually in contact with the sperm. The Sertoli cells are also going to be target cells for a bunch of hormones. Specifically, they're target cells for testosterone, right? So testosterone is going to actively act on Sertoli cells to stimulate spermatogenesis. So the sperm, we're going to see in a minute, are going to be kind of developing here in this area between the Sertoli cells. So testosterone is acting on that process, allowing the Sertoli cells to help these sperm develop. And FSH, follicle stimulating hormone, which we talked about a long time ago when we talked about the endocrine system and we talked about the hypothalamus. It was part of one of our flows of hormones, right, our tropic hormones. They come back into play now. They're also acting on the Sertoli cells, helping stimulate spermatogenesis, helping stimulate the development of sperm. The Sertoli cells do have their own hormone that they secrete. So they don't secrete testosterone, they don't secrete FSH. What they do secrete is inhibin, which is a negative feedback loop on FSH. So once they're producing a bunch of sperm, they need to go back and tell FSH, hey, we made them, you can chill. That's what inhibin does. Okay. So once the sperm have been produced, they're going to travel through the epididymis, they're going to travel through the vas deferens, they're going to travel through the urethra, and the urethra is going to be going through the penis. So the penis is the male copulatory organ, and it's specialized for sperm deposition. So it's specialized for allowing us to get the sperm close and in contact with ova, with egg cells from the female reproductive system. So as we move through and we think about hormonal and neural control, we're also going to want to think, with respect to the male reproductive system, about erection, ejaculation. That's the end part of this process. So at rest, the penis is flaccid. So flaccid. The erectile tissue don't have that much blood in them in this state, which is why we see this flaccidity, which is why we see this laxity, right? This relaxed state. But during sexual arousal, we have blood flow to the penis increase. Blood flow enters the erectile tissues, which are the corpora cavernosa and the corpus spongiosum that you looked at mostly in cross-section in lab. And those tissues are going to fill with blood. They're going to fill with blood and they're going to swell with blood. And the blood flow is going to be kind of blocked from returning to the body, which causes erection. So that's what causes the penis to harden and lengthen. It's because this blood is filling the penis with volume. Okay. And this happens, and then we have mechanical stimulation until ejaculation occurs, which is when the sperm gets ejected through the urethra, again, due to muscle contractions, basically, both skeletal and smooth. And then the penis would return to its flaccid state. The blood flow would be allowed to return to the body, which is good. You don't want to have a 4-hour erection, right? I think I've seen ads saying, go to the hospital, right? So this would return to its normal state afterwards. And we'll talk a bit about the neural control of that. In terms of where the testes are, where we're talking about this sperm development, the testes are located in the scrotum, which is really what we think of as the external genitalia, so or at least part of the external genitalia of the male reproductive system. So the scrotum is suspended beneath the penis, houses the testes, and it's there, it's external, largely because sperm can't develop at body temperature. So we'll see in the female reproductive system, ovaries and the entire female reproductive tract are located within the pelvis, but sperm development has to happen at a lower body temperature. So the testes, which are producing the sperm, have to be housed outside of the body so that they can be a little cooler. So this is why you'll sometimes see articles about how wearing pants that are too tight or constantly having your laptop in your lap can damage sperm. It's because of these effects on temperature. It's because this physical positioning is deliberately there to allow for cooling. So the pathway for our sperm goes through the seminiferous tubules, where they're really being produced and placed in the lumen. And then through peristalsis, those seminiferous tubules are going to kind of squeeze the sperm along. They're going to go through structures called the rete testes and the efferent ductules, which are just more tubes that allow the sperm to exit the testes, where they go into the epididymis, which we saw has a head, a body, and a tail in lab, and ends in the vas deferens. So that's just kind of the pathway the sperm are taking as they exit the testes. As the sperm are exiting, they're going through the vas deferens. The vas deferens then goes into the inguinal canal. It's passing kind of behind and under the bladder. It's going to pass by our accessory glands. So there are 3 of them that we're interested in. I kind of pointed them out when we were looking at that picture of the male reproductive tract, but they are the seminal vesicles, the prostate gland, and the bulbourethral glands. And they each have their own job to do. The most interesting one to me is the seminal vesicles. So the seminal vesicles are producing about 70% of the contents of semen. So they're producing an alkaline fluid that is mixing with the sperm. So the sperm are coming from the testes, but the fluid component of semen is really coming from these glands. So the fluid produced by the seminal vesicles is alkaline, largely because the female reproductive tract is slightly acidic. So this is to kind of neutralize the acidity in the female reproductive tract. It also includes fructose, enzymes, prostaglandins that also allow the sperm to have better motility, better ability to fertilize eggs. So they're feeding the sperm. They're helping the sperm do their job, helping the sperm break down the outer layer of the ova, things like that. So they have a big job to do, even though they're not producing sperm themselves. The prostate gland secretes something called citrate and some more enzymes. And actually the function exactly of the citrate and the enzymes is not very well understood, but they are contributing like 20%, a bit more, to the contents of the semen. And then we pass by the bulbourethral glands, which are quite small. What they're secreting is a viscous fluid that contains a lot of mucus. And this is mainly for lubrication. Here's a table from your book about the actions of androgens in males. So mainly we're thinking about testosterone. So they're going to stimulate spermatogenesis, which is a lot of our focus today. They're going to promote development of secondary sex characteristics. They're going to increase sex drive. So testosterone is a lot of what produces sex drive. They promote protein synthesis in skeletal muscle. So this is one of the reasons that males are generally more muscular than females, is because of this testosterone. They also stimulate the secretion of growth hormone, which allows for growth spurts, allows for bone growth in adolescence. And then they, as we've covered, they promote the development of the male reproductive structures in the first place during embryology, during embryonic life. Okay. So we want to look at the development of a sperm, and that means we also want to know what a mature sperm looks like, what are its parts. So the sperm can be divided into kind of 3 pieces. It has a head, so this top part, it has a mid piece, and it has a tail. Okay. So each piece of the mature sperm has its own job to do. So the head is largely the receptacle for carrying chromosomes, so carrying the genetic information. So the nucleus of the sperm, remember sperm like are a cell. So the nucleus of the sperm is located here in the head. So this is where we'd see our chromosomes. Right? These are my chromosomes, my great drawer. The top of the head of the sperm has another important piece, something called the acrosome. So the acrosome contains enzymes that are going to allow the sperm to break into the ova in order to deposit their genetic information during sexual reproduction. So those enzymes in the acrosome are necessary for fertilization. The other 2 parts of the sperm are really related to the fact that the sperm needs to move, like it needs to move through the reproductive tract, the male reproductive tract. We know that's helped along by peristalsis, but then it needs to swim through the female reproductive tract. So it has a lot of mitochondria. Those are located in the midpiece. So that's what these red circles are in this image. And then it has a tail. So this tail makes whip-like movements, and as it thrashes back and forth, that propels the sperm forward. So now we're going to look at the development of a sperm. So how we get from a normal diploid cell in the testes to these mature sperm, which are going to be haploid and are going to have these specialized structures for movement through the reproductive system. Okay. So the cells we start out with are called spermatogonia. So spermatogonia are in the gonads, are in the testes, but we want to note here, they are diploid cells. That's what 2N means, which means that they have the full normal number of chromosomes like we see everywhere else in the body. So they have 2 sets of 23 chromosomes for a total of 46. Okay. So spermatogonia can reproduce just like any other cells reproduce in your body. So they can undergo mitosis and make more spermatogonia. So we're going to see them located kind of on the outside of our seminiferous tubules. So out in this kind of basal area of our Sertoli cells. So these are 2 Sertoli cells, and we're seeing the sperm development in this kind of canal between them. So we start out with our spermatogonia, and as we move towards the development of sperm, we're going to take the spermatogonia and they are going to undergo meiosis. So we're going to have the spermatogonia. We're going to have one kind of start to descend between the Sertoli cells, producing our primary spermatocytes. Primary spermatocytes. So we would have 2 of them, only one is drawn here. We are then going to produce our secondary spermatocytes after meiosis 1. So remember, meiosis is a specialized form of cell division that occurs in order to produce gametes, so in order to produce sperm. And it's basically we have a cell divide in half, and then we have those 2 new cells divide in half again without any replication of the genetic material. Okay, so that's what's going on here. The first split is happening to give us our secondary spermatocytes. And then our second split gives us our spermatids. So the spermatids are really already down to the amount of genetic material we want to deposit. So these are haploid cells. So N means they're haploid, means they only have 1 set of chromosomes. So at this point, they only have 23 chromosomes. Each gene that they have only has 1 allele at this point. But the spermatids aren't completely sperm yet. They're not spermatozoa. That's because to be technically termed spermatozoa, we need to have that physical structure that we just went over. We need to have this head, midpiece, and tail. So the spermatids grow into this structure, producing the head kind of around their nucleus, and stretching out and producing the midpiece and tail, kind of moving down this image. Okay. So in terms of what I think it's important for you to know from this slide, because I know this is a bunch of terms, and specifically because we're not talking much about mitosis and meiosis today, this might be a little bit of a shock that you need to remember those processes. So I want you to know spermatogonia are the cells that we start with, and they're diploid. Okay. So diploid is that 2N. Then I want you to know they undergo meiosis and become spermatids, which are haploid. And then as they develop their head, midpiece, and tail form, we're going to refer to them as spermatozoa, and that's different from a spermatid. because of that physical structure. Okay. So the rest of these kind of numbers on this slide, it's okay if you don't go too into depth on those, but no, we start with spermatogonia. They become spermatids, which only have half the genetic information, and then they develop into spermatozoa when they develop their structures that are going to allow them to move through the reproductive system and fertilize an egg. Okay. So this is just happening between the Sertoli cells. So it's happening from the basement membrane, which was out kind of at the edge of the Sertoli cells, to the lumen, which is the center of that tubule. Okay. As this happens, the spermatogonia, which were those cells that we start with, one of them is going to stick back so that we can produce more spermatogonia, so that we can produce more sperm in the future. The spermatozoa, which are a final product, get released into the lumen of the seminiferous tubules, and they actually stay there immotile, so not moving, for 20 days. So they're not produced immediately right before release. They have to hang out for a bit, and they are going to have to undergo a little more processing to be considered fully mature sperm. Once they are ready for release, they're going to move through the epididymis by peristalsis and also by the flow of fluid in the lumen. And then they're going to move to the vas deferens by peristalsis, and they're going to stay there in the vas deferens until ejaculation. So that leads us to the process that leads up to ejaculation, so the sexual response in males. So rather than being controlled by testosterone, by the hormones we've been talking about for developing sperm, the actual sexual response during intercourse, during copulation, is controlled by the nervous system. So it's specifically controlled by the autonomic nervous system. So we're going to be thinking parasympathetic, sympathetic is what we should be expecting to be looking at as we're thinking about the sexual response. Yeah. So it's actually a spinal reflex. So the nerves are relatively close to this pelvic region, but they take their normal terms for the autonomic nervous system like we saw elsewhere in the body. Okay. So the sexual response has 3 phases. The first is erection, where we have blood flow to the tissue. Now, erection is controlled by the parasympathetic nervous system. And then we have emission, which is the movement of the sperm and semen into the urethra. This is going to be controlled by the sympathetic nervous system. And then we have ejaculation, which differs from emission in that ejaculation is the actual release of the semen to the outside environment, but it's also controlled by the sympathetic nervous system. So the only one that's parasympathetic really is the initial response, the initial erection. And the stimuli for the sexual response, this is going to be controlled by mechanoreceptors of the penis and other erogenous zones, so sensations of physical touch, as well as some neural input from the brain, so what you're thinking, what you're seeing, what you're feeling. Okay. So some of this, so that's the part that's descending from higher brain centers. Okay. So the flow of this process would look something like this. We begin with mechanical stimulation and your thoughts, right? So activity coming from neural pathways in your brain. This impulse comes to your spinal cord. So in the spinal cord, what happens is that we have lowered sympathetic activity in the neurons to the penis, and we're raising the parasympathetic activity, which is what's causing the erection. So specifically what happens is that the arterioles supplying the blood to the erectile tissue, the smooth muscle around them relaxes, so they dilate, so they have greater flow. As more blood flows into the penis, it produces pressure and volume. The pressure and volume itself are the erection, Another thing that's happening at the same time is because we're in this really tight area, right? We're in the erectile tissue. There's only so much it can expand. So as these arterioles expand, this pushes on the veins and compresses them, which stops the outflow of blood, which means that the volume and pressure are increasing even more in the erectile tissue, further creating the development of an erection. Okay, so we will pick up here on Friday, which is our last bit of new material.